Hello everyone, this is your pre-lab lecture for EAS 1600, Lab 10, the carbon cycle. As you can see here in these beautiful pictures, carbon comes in many different flavors, but for the purposes of our lab and really when we're talking about the carbon cycle in general, we're really just concerned with breaking down carbon into two different groups, and that which is organic versus that which is inorganic. So the carbon that contains these carbon-carbon and carbon-hydrogen bonds, versus carbon that lacks these bonds and instead is usually bonded to oxygen such as CO2, carbon dioxide. So here's an overview of the carbon cycle from the Department of Energy. This cartoon was produced back in 2008, so it's a little old but still very, very relevant. And we have our major reservoirs in white right here, so the atmosphere, for example, and in parentheses would be the mass of carbon that's held in this reservoir, so in this case 800 gigatons of carbon. And then we have these fluxes in white, and so this would be gigatons per year. And then in yellow we have these natural processes, which are usually, or in this case mostly, short-term cycling. And the carbon cycle in general is definitely driven by these two different types of cycles. So our long term, we're talking about burial and sedimentation. And so here we have the fossil pool, which would represent one of these reservoirs, versus our short term cycling. So soil respiration, microbial respiration and decomposition, plant respiration, so on and so forth. And then in red right here, we have our own human inputs or anthropogenic inputs. So we can always be mindful of that because as we're going through the lab, we're going to see if some of these fluxes are in steady state. That is, does this flux in to the atmosphere, for example, equal the flux that's leaving it? Does that equal zero? Or the difference between them equal zero, which would imply an equilibrium or a steady state condition. And so again, we have these carbon fluxes, and this is normally expressed in terms of gigatons carbon per year, missing that year right there, so remember that. And to define the flux, it's the amount of carbon that's exchanged per year or per time between these different reservoirs. And these reservoirs will include inorganic and organic carbon, but when we're talking about the fluxes of carbon themselves, we're usually just talking about carbon dioxide. And we can calculate the residence time. We can find out how long a mole of carbon, for example, will remain in one of these reservoirs. But how do we do that? So the residence time is the average time that's spent in a reservoir of a certain molecule. In our case, we're talking about carbon, so CO2, or just the carbon part of the CO2 molecule. And mathematically, we can express this as the reservoir size over the flux in or out. We have to define that. So reservoir size in gigatons over the flux, which is expressed in gigatons per year. The gigatons cancel and we're left with years. And this diagram right here, this is from your lab. And here it was kind of nice. It breaks it down between the long-term cycling and the red and blue. And then in red, we have the short-term cycling. So long-term carbonate sediments, for example, are organic, and red represented by the exchange between the surface ocean and the deep ocean, for example. Well, let's work through an exercise. So go ahead and take a moment to calculate the residence time of carbon in the surface ocean reservoir. So this box right here. And you can go ahead and pause the video to give yourself some time to work through this. Think about what two pieces of information you need, which we can get from this equation right here. So go ahead and take a minute or two to work this out. All right, so you've resumed the video and presumably you've at least tried working this out. And so we need to know the reservoir size, and that's clearly given to you right there inside that box, surface ocean reservoir, in this case 1020 gigatons. And then we're going to calculate the flux out. And we're only concerned with the flux out because we're talking about, on average, a mole of carbon that enters the system and how long it's there before it leaves. So that's why we just want the flux out and we're forgetting about the flux in. And so these arrows, these vectors represent the fluxes. And so anything that's pointing away would be a flux out. And so we simply just sum 92 right here in the air-ocean exchange with 91.6 gigatons per year from the exchange between the surface ocean and the deep ocean. And that yields 183.6 gigatons per year. So tau, the Greek letter tau here representing residence time, would equal that reservoir value. So 1020 gigatons over the 183.6 gigatons per year, and we're left with 5.6 years for our residence time. All right, but are we in a steady state? In other words, does our flux in equal our flux out? So these arrows out, do they equal the ones that are going in? So let's go ahead and take a look at that. And so we can calculate the flux in by just summing up these arrows, these values next to the arrows that are pointing in towards the surface ocean reservoir. So 100 plus 90, 190 gigatons per year minus the value that we already calculated for the flux out, 183.6 gigatons per year, and that yields a positive 6.4 gigatons per year. So because this value is positive, we know that there's more carbon that's fluxing in than is leaving that reservoir. So over time, the surface ocean, in this case, is accumulating carbon. All right, so let's move to the lab itself and give you some hints and guidance. And this pertains to the questions 6 through 15. So in this part of the experiment, we have the experimental setup. We have a sink that's filled with water, represented right here in the green box. And then we have a graduated cylinder that represents a water column, and that's up on the bench. Now we have a peristaltic pump that's connected to the sink. In our workflow, we'd have the tubing go from the sink, so the inflow to the pump, and then the pump's outflow to that water column right there. And we're going to have a flow of water that goes from the sink through the pump and into the water column. Now this water column, or this graduated cylinder, has holes towards the top. And so as water fills up and reaches these holes, the water will begin to flow out from the column and back into the sink. So mind you, there's always water in the sink. The sink is plugged and it's not draining. All right. And in this experiment, this diagram, we have a simplified carbon cycle. And so we have a sink, which represents the land plus sea reservoirs. And then we have the column, which represents the atmosphere reservoir for carbon. Water is the amount of carbon. It represents the amount of carbon measured in grams. And the flow of water itself is the actual flux of CO2. And we're going to measure this in grams per second. So when we turn the pump on, we're going to draw water from the sink through the pump and into the water column. The column is going to fill up. And as we reach these holes again, water will begin to flow out. Now at some point, we'll reach an equilibrium volume where the flux in equals the flux out. And we're going to convert that volume to mass to calculate the reservoir value in grams. And so we can actually plot the reservoir size as we're filling up this water column over time. So here we have reservoir size in grams over time. And we want to ask ourselves the question, is that rate of change of the column mass, that is, the atmospheric carbon reservoir, constant? Well, as it is initially filling, it is constant, right? As we can see by this straight line. But as we reach the holes towards the top of the column, some of that water is leaving that system, leaving that reservoir. So now we don't have this constant rate. Now it's changing, right? And from the shape of this curve, you can take a step back and you can infer what the steady state mass actually is. And that's one of your questions on the lab. Okay, so pretty straightforward. A lot of work you don't have to do. We're giving you a lot of the data, and I know in the lab that that's half the battle. So that's all I really have for you in this pre-lab lecture. Now mind you, I'll be available during your regular scheduled time for any questions that you have. We'll use BlueJeans. There's a BlueJeans link within Canvas, and I'll also send out a separate email for you so you can have that. Now, if you're good to go with and you don't have any questions or anything, you don't have to log on. But if you just want to be a part of the group and sort of be there and have this time as your dedicated time to work in the lab, to ask questions, please, by all means, log on. You can ask questions either verbally or just through the chat window so that everyone can see. And maybe I'll share my screen here and there to guide some of you if I see a trend or a pattern in certain questions. And worst case, if you have issues on your end and need to share your screen, you can go ahead and do that as well. I hope this goes okay for everyone, but please, by all means, provide us some feedback so we can work together to improve these different lectures. These are some weird times that we're in. None of us have really done this before, so we're kind of all learning on the fly. So good luck with the lab, and let me know if you have any questions.